Okay, we continue with our immunology class, and today we want to look at T cells and the role that they play in cell-mediated immunity. So let's just have an idea of what these two terms mean. So we have two terms, T cells, then the other term is cell-mediated immunity. So T cells, or otherwise referred to as T lymphocytes, they are a type of white blood cells who play a major role in protecting our bodies against disease. They are particularly significant when you have an infection that is inside your body cells, because when the infection is inside your body cells, then the antibodies cannot reach such an infection easily. Okay? So T cells happen to play a central role to what is called cell-mediated immunity. So what is this cell-mediated immunity then? This refers to a branch of the immune system which does not depend on antibodies, but relies mainly on the use of T cells. And by doing so, it targets infections that are found living inside our body cells. So it means when there is an infection inside the cell, then antibodies won't reach that infection. So then the immune system has to apply a different mechanism to target the pathogen. In this case, it now brings in the cell-mediated immunity, which is run entirely by T cells. Okay, so antibodies cannot reach when pathogens are found inside our body cells. There has to be something else that is used, and in this case, the T cells are called into action. So why is this important? Why is cell-mediated immunity important? There are some pathogens which hide or which live and continue to multiply inside cells. Such pathogens, for example, are viruses that cause HIV. Okay, so the human immunodeficiency virus, that is such an example. You have some intracellular bacteria that live inside a cell. For example, the Mycobacterium tuberculosis, the one that is responsible for causing TB, at least in humans. Then another example are the cancerous cells, okay? The cancer cells, the tumor cells. So because these pathogens are inside cells, then it means antibodies which are found circulating in the body fluids, the fluids which are found in between cells but not inside the cell. Okay, so it means whatever is contained in the fluid cannot reach where the pathogens are inside the cells. Then it means these antibodies cannot therefore destroy these pathogens that are found living and multiplying inside our cells. So to deal with this, then the immune system now employs a different mechanism. In this case, the T cells. It uses the T cells to detect these infected cells. Then after detecting them, it destroys those cells. So it has to detect first so that it does not destroy what is not infected. Then after that destruction, because the cell that's infected has been destroyed, then it means the infection will not be able to spread, because there has been a destruction of those cells that are infected. What is important to note is that the antibodies cannot do this, because the infection, the pathogen inapatikana ndani ya the cell. So some other mechanism, in this case the T cells, they have now to do that particular role. So humoral immunity is different from cell-mediated immunity in several ways. The cells that play the main role between the two types or branches of immunity are different. T cells lead cell-mediated immunity, while the B cells lead the humoral immunity. B cells are the ones that lead to the production of antibodies. Okay? So the use of antibodies, cell-mediated immunity does not use antibodies. But humoral immunity uses antibodies which are produced by the B cells. The target, cell-mediated immunity targets the infected body cells. It targets the cell. But the humoral immunity, on the other hand, it targets the free pathogen which is found in the body fluid. Okay, so the targets are different. Cell-mediated immunity, driven by T cells, targets a cell which has an infection inside, which has a pathogen inside it. But the humoral immunity, driven by B cells, which produce antibodies, targets pathogens which are found circulating in the body fluids. Cell-mediated immunity works best against pathogens which live inside cells. So things like viruses, the intracellular bacteria like the Mycobacterium TB, and other cancerous cells. Okay. But the humoral immunity, on the other hand, works best against these pathogens found in fluids. Example, bacteria, toxins, and other extracellular pathogens. Humoral immunity is not targeting pathogens that are found inside cells. Instead, it is targeting those pathogens that are found outside the cells. Okay? So it means then, for there to be proper protection, cell-mediated immunity needs to work together with the humoral immunity. The only difference is that they have specialized in different types of threats. Cell-mediated immunity looks at the inside of cells. Humoral immunity looks at the fluid that is circulating within the body. So they are looking for pathogens, but at different places within the body. Okay? So we've seen that T cells help with the cell-mediated immunity. But where do these T cells come from? So T cells originate from the bone marrow. Okay? They begin as immature cells in the bone marrow. But because they are still immature, it means at that stage within the bone marrow, they are not yet functional. So they need to mature. Okay? So their process of maturing happens in the thymus gland. The thymus gland is located just above the heart. Okay, so while in the thymus, these T cells, which were produced in the bone marrow, okay, they move to the thymus gland, then they begin to mature. And as they mature, they develop some specific receptors. These are receptors which are specific for the T cells, so we call them T cell receptors. So within the thymus gland, the T cells mature. And in the process of maturation, they also develop the T cell receptors. Another thing that happens within the thymus gland for the T cells is they learn how to distinguish between the normal body cells and the foreign antigens. So they learn to distinguish between self and non-self. That process of distinguishing between self and non-self is a very important stage in immunology, because if T cells fail to recognize a normal body cell from a foreign antigen, then they will end up attacking the body's own cells and tissues. Once this happens, then we end up with what we call autoimmune diseases. So the immune system fights against its own body cells. Okay? So it's fighting against its own body cells because it does not have the ability to detect what is self from what is non-self. Haiwezi tofautisha kati ya the cells that belong to your body and what is foreign to your body. So then it means it attacks your normal body cells the same way it will attack a foreign antigen. So with that, you now end up developing diseases as a result of that immune reaction. But because it's an immune reaction against your own body cells, we now call such diseases autoimmune. Okay? Autoimmune diseases. So, if, for example, the T cells have to distinguish between your own body cells and something that is foreign, okay? Something that is not belonging to your body cell, then it means there has to be a way in which these T cells now recognize a cell that is infected from a cell that is not infected. There has to be something that the T cell uses to differentiate between a normal cell and an infected cell. So what is clear is that the T cells do not recognize whole pathogens directly. It does not recognize a virus as it is. It does not recognize an intracellular bacteria as it is. Instead, once a cell is infected, that cell, upon infection, will display small pieces of the pathogen. Those small pieces, what we call antigens, will be displayed on the surface of the infected cell. Then, those antigens that are displayed, those antigens that are on the surface, okay, are then presented by special molecules. We call it the major histocompatibility complex proteins. So you have an infected cell. That infected cell, on the surface of that cell, there will be antigens, pieces of the pathogen which has infected that cell. Okay? on the surface of that cell. The antigens are on the surface. Those antigens belong to the pathogen. They are not part of your body. Okay? Then those antigens are now presented by special molecules, what we call the MHC proteins. So T cells will now use their T cell receptors to recognize the combination of the antigen on the surface of the infected cell and the MHC protein, because it is now that protein, the MHC, which has to present. But these antigens will already be on the cell which has the pathogen inside it. Okay? So it means the T cell cannot recognize an entire pathogen. The T cell will recognize that there is this particular pathogen inside this cell. If there is a component of that pathogen, what we are calling an antigen, being displayed, okay, on the surface of that cell. But for the T cell to know, for the T cell to recognize that antigen, there has to be another special molecule which has to present that antigen to the T cell receptor. So for the T cell receptor to know, to recognize, it needs the antigen from the pathogen, which has now to combine with a major histocompatibility complex protein before now the T cell can recognize it. Okay? So it means if the pathogen is in a cell, that pathogen will have its antigens, pieces, small pieces of that pathogen on the surface of that cell. Then the MHC proteins will come and combine with these antigens. Then the MHC protein will now present those antigens to the T cell receptor. So T cell receptor only recognizes these antigens after they have combined with the MHC proteins. Okay? So upon combining the MHC protein with the antigen, which will be on the surface of the infected cell, then now the T cell receptor can recognize that cell as potentially being either infected or abnormal. Abnormal in the case of cancer, for example, but infected if there is a virus inside there, or if there is an intracellular bacteria. So if, for example, there is HIV infected cell, that cell will have antigens of the HIV virus on its surface. Then the MHC protein will now bind to those antigen on the surface of that cell, the HIV antigens on the surface of that infected cell. So the MHC proteins, upon binding to that antigen, will then allow the T cell receptors, which now the T cells is using to recognize that particular antigen MHC combination. So you can think of the MHC antigen combination as a flag. Okay? Fikiria moja ni kama pole na ingine ikuwe ni kama flag. Huwezi ona flag ikipepea juu kama haijawekwa kwa pole. Okay? Ukitoa pole, flag itaanguka chini. But ukisimamisha pole bila flag, hutaweza kuona flag. So for you to see the flag up, it must have a pole. Then on that pole now, you add the flag so that you can see that flag being displayed high. Okay, so that is how the T cells end up recognizing a cell that is either infected or abnormal. It looks out for the small pieces of that antigen, of that pathogen, which we are calling an antigen. And those small pieces will be displayed on the surface of that infected cell. But those antigens have to be presented by a special molecule which we call the MHC protein. So the MHC protein combines with the antigen before it can now be presented and recognized by the T cell using the T cell receptors. Okay, so without the MHC protein, then the antigen cannot be recognized by the T cell receptors. So what are the main functions of these T cells? The first is recognizing an infected cell or an abnormal cell. Okay? So they continuously circulate in the blood. Those are the T cells, either in the blood or in the lymphatic system. Then using the antigen and the MHC protein, they will continuously be able to detect any cell that has an infection, or any cell that is abnormal, or any cell that is foreign, something that has been transplanted. Okay. The other role that the T cells do is they destruction of the infected T cells. So there are special T cells, the cytotoxic T cells, which will directly kill a cell that is infected or a cell that is abnormal. It will kill directly. How does these cytotoxic T cells, how do they manage to either kill an infected cell or kill an abnormal cell? They are able to kill, they are able to destroy infected or abnormal cells in around 3 ways. One, they do it by releasing toxic proteins. Okay? They release proteins that are toxic. Then now these toxic proteins are the ones that will end up killing either the cell which is infected or the cell which is abnormal. These toxic proteins are mainly of two types, the perforins and the granzymes. The other way in which the cytotoxic T cells can destroy either a cell which is infected or a cell which is abnormal is by creating pores in the cell membrane of whichever cell that is either infected or abnormal. Inaweka mashimo on the cell membrane of that particular cell. Okay, so because of those pores, then the contents of that cell will pour out. Then the cell will not manage to survive, but it will die. The last way is when it does what we call apoptosis. Apoptosis is programmed cell death. So the T cells, the cytotoxic T cells, they can trigger apoptosis. They can lead to apoptosis. So by doing this, by destroying a cell that is either infected or abnormal, then these T cells will help to prevent the pathogens which are found inside that cell from spreading, from multiplying to other cells, because it has led to the destruction of that infected cell. So it means the pathogen will also die in the process. Okay. Another thing is the T cells help in the regulation of immune responses. So some T cells will help in the controlling how the immune system responds, so that it regulates how the immune system responds in order to prevent the immune system from damaging tissues that are healthy. Okay? So these T cells that help in the regulation of the immune response, they do that by either activating other immune cells when needed. When they need the immune system to be hyped, they activate other immune cells. When they need the immune system to reduce in its activity, then they have to suppress the immune reactions when the reactions are now excessive. Because if it is not suppressed, then it can lead to the destruction of the healthy body tissues. Okay? Ni kama unapika kitu. Ukitaka kuongeza moto, inakuongezea. Ukitaka kupunguza, inapunguza. Okay? Because when it is needed that the immune system should be activated more, these T cells do that. When it's needed that the immune system has to be, the immune response, the immune reaction has to be suppressed, ipunguzwe, again, these T cells do that. So if this regulation is not done, then the immune system could end up leading to very serious tissue damages. Okay? So if you don't suppress the immune reaction when it is becoming excessive, ni kama unachoma ama unapika. Chakula ikifika mahali hivi lazima uzime moto, because usipozima hiyo moto itaunguisha chakula. But inafika mahali unaona hii moto haijatosha, unaongeza. Okay, so the T cells are just helping in the regulation. When it's needed to increase, it increases. When it's needed in suppression of the reaction, it suppresses. Okay. So those are the three main functions or main roles that are performed by T cells. But what exactly are the types of the T cells that are present in any immune system? So the first are the helper T cells, or the CD4 cells. Okay. So the helper T cells, their function is to coordinate the immune response. They activate B cells, macrophages, and cytotoxic T cells. They activate B cells, then B cells will lead to the production of what? Antibodies. Macrophages, kazi yake is to swallow. Okay, to phagocytose. Hiyo ni kazi ya macrophage. Then cytotoxic T cells, kazi yake ni kufanya nini? Ni kuua directly. Okay. So the B cells, those are responsible for humoral immunity because they lead to the production of what? Antibodies. Cytotoxic T cells, just like any other T cell, that is responsible for the cell-mediated immunity. So it means that the helper T cells end up being critical T cells, not only for the cell-mediated immunity, but also for the humoral immunity. Okay. So what is important is some viral infections, for example HIV, they target these CD4 cells. Okay. So once these CD4 cells have been targeted by the HIV virus, then they will decrease in number because they will be killed by some other type of cell which I'll talk about next. And once the CD4 cells have reduced, then they will not be able to coordinate the immune response. A reduction in that coordination, then a reduction in the activation of B cells, activation of macrophages, and activation of cytotoxic T cells means that once the CD4 cells have reduced or the helper T cells have been targeted and reduced, then the immunity will be weakened. The second type are the CD8 or the cytotoxic T cells. What the CD8 do, these are the ones that now directly kill cells that are either infected or abnormal. Okay. So these are the ones that will kill a CD4 cell if it has been infected, targeted by HIV. Okay. So because they kill cells that are infected, mostly cells that are infected will be infected by viruses or intracellular bacteria like TB, okay, or abnormal cells in terms of cancer. So it means the cytotoxic T cells or the CD8 T cells, they are key players in the immunity against viruses because they are targeting and killing cells that are infected. What lives inside a cell mostly are viruses. Okay. The third type of T cell are the ones that are responsible for the suppression. The third type, sorry about that, that third type is about a group of T cells which are responsible in the suppression of the immune responses whenever these responses are excessive. Okay, so we call them the regulatory T cells. The regulatory T cells. So just from that word, it means they do some regulation. When the response is too much, is excess, it has to be regulated. It has to be brought down. Okay? So because they reduce the excessiveness of these immune responses, then it means these regulatory T cells are critical in the prevention of autoimmune diseases. The fourth type of T cells is what we call the memory T cell. After any infection, there are some T cells that will remain in the body. Okay? These T cells are remaining there and will help in providing faster and stronger response during a future similar infection. Okay? So if there was an infection, for example, for any pathogen, think of it like HIV, for example, then the cell led to the production of, that infection led to the production of several T cells. After that infection, not all those T cells will be destroyed. There are some T cells that will remain inside your body. The importance of that is in case there will be a similar infection, a similar attack in the future, then those memory T cells which were kept in your body, which were retained in your body, will help in providing not only a response which is faster but stronger. Okay? And this is now mainly how vaccines work. Okay? Because with a vaccine, you are giving someone a pathogen which has been attenuated. That pathogen leads to the production of T cells. Some of those T cells will not be destroyed. They will be kept. They will remain in the body. So the importance of activating those T cells, then they are kept in memory as memory T cells. In case now you are infected with the real pathogen, the one which is not attenuated, then these T cells are brought in faster to help mop up that infection. So that's the importance and that's the principle on which vaccines work. The importance of memory T cells is providing a faster and stronger response in case there is a future similar attack. That is the principle upon which vaccines work. Okay? So is cell-mediated immunity crucial? Okay? The answer is yes. And why is it so? It's because there are many pathogens which are found multiplying inside our cells. Equally, we have abnormal cells as a result of cancer. So if we didn't have T cells, which are the main players in this cell-mediated immunity, then the viral infections are found inside our cells. Those viral infections will spread rapidly. Then intracellular bacterial infections, specifically like Mycobacterium tuberculosis, which causes TB, they will be difficult to control. And the abnormal cells, for example, during cancer, it will be difficult to deal with them. Why? Because antibodies don't have the ability to reach inside our cells. So it means any intracellular infection will not be possible to be cleared had it not been for the work of T cells. Okay? So, in summary, T cells are white blood cells, and they originate in the bone marrow, but they mature in the thymus. We said the thymus gland is just located slightly above our heart. Okay. As they mature in the thymus, they are given that ability to recognize to differentiate between self and non-self cells. Okay. Then they have the ability, these T cells, to recognize a cell that is infected and a cell that is abnormal. That recognition is enabled by the combination of the antigen, which is a product of the infectious agent, and that antigen will be found on the surface of the cell where the pathogen has infected. Then that antigen, for it to be recognized by the T cell, it has to be presented by special proteins. The proteins that present the antigen are major histocompatibility complex proteins. So it means for the T cell to recognize an antigen, that antigen must be joined and presented by an MHC protein. Okay. The T cells have an ability to destroy these cells that are infected, and also destroying the abnormal cells in the case of cancer, for example. Okay. There are some T cells as well which will help in the regulation of the immune system. Okay? The regulatory T cells. The T cells that destroy infected cells, those are called the cytotoxic T cells. Then, there are some T cells that after an infection, they will remain in the body. The importance of those T cells remaining in the body is they provide long-term immune memory. So that in case a similar pathogen infects again, a similar pathogen strikes again, then these T cells are mobilized faster so that they can deal with that pathogen. And it is on the basis of that, on the memory T cells, that we have vaccines being produced and working. Okay. So cell-mediated immunity is antibody independent. It does not use antibodies. Instead, it uses T cells. And this cell-mediated immunity is important for eliminating virus-infected cells. A cell that has a virus inside it, the antibody cannot reach inside of such cells. Then we have bacteria which live inside cells, like the Mycobacterium tuberculosis. Then we have abnormal cells, for example, the cells that have the cancer cells. That is an abnormal cell, which has to be eliminated on the basis of a T cell, recognizing the abnormal nature of that cancerous cell. So in short, to conclude all this, to sum it up, antibodies help in fighting pathogens that are found outside cells. But T cells help to fight infections that are hiding inside cells. Okay? So ni kama, if you think of, you have a security patrol. Kuna wale wanaangalia ndani, na kuna wengine wanaangalia nje. So antibodies wanaangalia nje. Then T cells wanaangalia ndani. So all of them are working for the protection of the human body against pathogens. The only difference is one of them is looking at the pathogens that are found outside the cells. Those are antibodies. That type of immunity is humoral immunity. Then the T cells are concerned more with the antigens, with the pathogens that are inside our cells. Okay? Cell-mediated immunity. So. Okay? Up to that level, we've covered T cells. So we move now next to finalize on a bit this. Then I'll look at exactly how these T cells work. Okay? We've looked at the T cells, we've looked at the types of T cells. But let's look at now how these T cells exactly work. We said there are some T cells that kill. So how do they kill? We said they produce toxic proteins. How do these toxic proteins exactly work? Okay. So just as a recap, the immune system has two major arms: the humoral immunity and the cell-mediated immunity. Humoral immunity uses antibodies, and antibodies are produced by B cells. Cell-mediated immunity uses T cells. T cells are important, are vital in fighting viruses, as we've seen, intracellular bacteria, cancer cells, and transplanted tissues. Okay. These T cells mature in the thymus, and that's why they have that T. Okay, T, because of the word thymus. We saw the major types of T cells, the helper T cell, the CD4 T cells, okay, whose main function is for coordination and controlling the immune response. These helper T cells, or the CD4 cells, they don't kill the infected cells themselves. But instead, when these CD4 cells are activated, they release chemicals, which are called cytokines. Okay, so CD4 cells, the helper T cells, when they are activated, they release cytokines. Okay, then these cytokines will now lead to activation of B cells. And then once B cells have been activated, they will produce antibodies. Okay. Then those antibodies will now lead to the activation of the CD8 cytotoxic T cells. These cytotoxic T cells are the ones that now kill the infected cells. The cytokines as well will activate macrophages. Macrophages kazi yake ni kumeza, is to engulf pathogens. But for them to engulf, ili kumeza the pathogens better, those macrophages have to be activated. How are they activated? By the production of what? Cytokines. Okay. We saw the cytotoxic T cells, or what we are calling the CD8. CD8 kazi yake ni kuua the infected cell or the abnormal cell. Okay. So they will kill a cell that has a pathogen inside it. Okay. They will also kill a cell that is abnormal. So they will identify a cell which is abnormal and a cell which has a pathogen inside it, and they will kill that cell. But how do the cytotoxic T cells kill these cells? They do kill by two main powerful molecules. The first one is the perforins. The perforins are the ones who have the ability to create holes in the infected cell membrane. Okay, so the cytotoxic T cells can kill an infected cell by using perforin. You can think of perforins and remember the word perforate. Okay, so perforins perforate the cell membrane of an infected cell. To perforate is to create a hole. Okay, then once they've created holes, then there are granzymes, another molecule, which will now enter through the holes that the perforins have created. Then once they are inside, they will activate cells inside that. They will activate enzymes inside that cell. Then those enzymes are the ones that will now lead to the death of that particular cell. Okay, so the granzymes enter through the holes that have been created by perforins. Once those enzymes, the granzymes, have gotten inside the cell through the holes, then they will activate other enzymes inside that cell to kill that particular cell. That is how now the cytotoxic T cells are actually killing the infected cells. The regulatory T cells, these are the ones that are like brakes. They are regulating, they are suppressing the immune response. Okay? It's important that the regulatory T cells work, because if the immune system becomes too active, it can damage normal tissues. It can cause autoimmune diseases. So these regulatory T cells help in reducing the immune activity. Okay? It helps in promoting and maintaining self-tolerance, so that the immune system can tolerate cells that belong to the body of that particular organism. So by doing that, then the regulatory T cells are helping to prevent the immune system from attacking itself. Okay? So your immune system cannot attack your own body because of regulatory T cells. If there were no regulatory T cells, then most likely the immune system will attack your own body. If the immune system attacks your own body, then it causes autoimmune diseases. Okay? The memory T cells, we said, these are like the record keepers. These are the ones that will hang around. Okay? These are the ones that will be able to identify the pathogen in the future. And because they've identified that pathogen in the future, then they can be mobilized faster to help in clearing that pathogen. So in essence, the memory T cells just help in providing long-term immunity. Okay? They help in providing long-term immunity. So that long-term immunity is provided because some of these memory T cells, they stay within the body for years. Within that time, they are able to recognize the same pathogen. So if you were attacked by Mycobacterium TB at some point, then your immune system cleared it. Some of the memory T cells will still hang around, so that in case the same bacteria attacks you in the future, the same T cells that helped will be mobilized so that they can respond faster and in a stronger way. Okay, so by doing that, then they deal with that particular pathogen. It is on the basis of these memory T cells that vaccines work. Then it's on the basis of these memory T cells that you end up noticing that when you have a second infection, it will mostly be mild. Haitapeleka mwili yako vibaya kama venye the first infection ilikupeleka. Because in your first infection, you didn't have memory T cells. But in your second infection, you now have memory T cells. And now they work, they respond quickly. Okay? So how exactly does the cell-mediated immunity work? Okay? So the first thing is that an antigen has to be presented. So before the T cells can act, remember cell-mediated immunity is immunity which is entirely being run by T cells. It's an arm of immunity that relies on T cells. So before these T cells can act, then the pathogen has to be recognized by the T cells. The T cells, as we saw, they cannot recognize the pathogen as a whole. So the pathogen has to be broken down into small pieces. These small pieces need to be antigens. Then these antigens, they now have to be presented, have to be displayed on the surface of that cell where the pathogen has infected. Okay? Whatever is displaying these antigens on the surface of that infected cell, that display is made possible by special molecules, which we call the major histocompatibility complex. This major histocompatibility complex, which helps in displaying the antigens on the surface of the infected cell, happens in two types. You have the MHC1 and the MHC2. So the MHC1, the major histocompatibility complex 1, is a type of MHC that is found on almost all cells. But it displays antigens from inside the cell. Then this MHC1 is recognized by the cytotoxic T cells. These are the CD8 T cells. Okay. So it means then the MHC class 1 is displaying antigens from inside the cell. So it means, in an easier way, the MHC1 functions by telling the immune system that something is wrong inside this cell. Hiyo ndio inatoa the antigens zenye ziko ndani ya hiyo cell iende kuzidisplay on the outside. Okay? So the MHC1 presents antigens that are from inside that particular cell. So it just tells the immune system that inside this cell I found antigens, and here they are. Okay? Then the MHC class 1 will be recognized by cytotoxic T cells that will now destroy that cell. Because by presenting antigens, it will mean that cell has been infected. So the cytotoxic T cells will now try to destroy that infected cell. How does the cytotoxic T cell destroy the cell? Use of perforin and granzymes. The perforins ni kufanya the perforations, kutengeneza mashimo on the cell membrane. the granzyme zitapitia kwa hiyo mashimo, ingia ndani ya cell, it will activate enzymes that will now lead to programmed cell death. The second type of MHC is the MHC class 2, the MHC type 2. The major histocompatibility type 2 is found only on the antigen-presenting cells. There are 3 antigen-presenting cells: the macrophages, dendritic cells, and B cells. Okay? So it means then the MHC class 2 is only capable of presenting antigens from pathogens which were swallowed. The dendritic cells, the macrophages, they engulf, okay, kumeza the pathogen. So once the pathogen has been engulfed by the macrophage, by the dendritic cell, then that engulfed pathogen will have its antigens, its broken down pieces displayed by the MHC class 2. Okay? So the MHC class 2 is working with the antigen presenting cells. What are the antigen presenting cells in this case? Macrophages and dendritic cells and B cells. What these do mostly is to engulf. Engulfing is surrounding a pathogen, swallowing that pathogen. Once the pathogen is engulfed, then antigens from that pathogen will now be displayed. Okay, will now be displayed because the pathogen has now been engulfed, has now been swallowed. So the MHC class 2 is mostly recognized by the CD4 helper T cells. Okay? So in a way, tuliona MHC class 1 inaambia immune system kwamba inside this cell there is something wrong. How does it do that? The MHC class 1 does that by presenting antigens that were found inside a cell. Why do we have antigens inside a cell? Because there was a pathogen inside that cell. The MHC class 2, hiyo inapatikana kwa antigen presenting cells, the APCs. 3 examples here: macrophages, dendritic cells, and B cells. Macrophages, they swallow, they engulf. Okay? So once they've engulfed, then the antigens of that engulfed pathogen will now be presented. Okay? They will now be presented. So the MHC class 2 then works by sort of telling the immune system that here I found a foreign invader. Because ni hiyo ipate the pathogen, then it surrounds the pathogen and now presents the antigen from that pathogen. Ni kama ufikirie MHC class 2 like a security provider mwenye anapata mwizi alafu anamshika na anaitana anasema mwizi ndio. Okay? But the MHC class 1 ni ule mwenye anapata mwizi na anaita anasema mwizi ako hapa ndani. Sawa sawa. The MHC class 2 anashika mwizi, anasema mwizi ndio huyu. Lakini the MHC class 1 anasema mwizi ako na ako ndani ya hii nyumba. Sawa sawa. The MHC class 1 is recognized by the CD8 cells. The CD8, those are the cytotoxic T cells. Once you've identified that there is an antigen from inside, it means there is a pathogen inside that cell. Cytotoxic T cells zitaua your cell. Okay? The CD4, ukisema there is a foreign invader, they will flag it. Then the cytotoxic T cells will eventually come and do what? And help in mopping. That is how the antigen recognition happens. It happens using the MHC class 1 and MHC class 2. MHC class 1 recognizes and presents an antigen from inside a cell. MHC class 2 recognizes and presents antigens after it has engulfed a pathogen. Okay. So a pathogen imekuwa immobilized, imekuwa engulfed, then MHC class 2 will now present the antigens from that immobilized, engulfed pathogen. So the second thing is now the T cell activation. So until T cells have recognized the antigen and the correct MHC molecule, they will always be inactive. Okay? The antigen and the MHC molecule, hizo ndio zinashikana before it can now be recognized by the T cell. So until zimeshikana, MHC class 1 kwa mfano imeshikana na an antigen ya particular virus. Okay? Ama imeshikana na antigen ya Mycobacterium TB. Ama imeshikana na particular antigen from an abnormal cell in terms of cancer. That is MHC class 1. Ikishikana na hiyo antigen, MHC class 1 with that antigen, then the T cell can now be activated and will now be able to recognize that antigen with the correct MHC molecule. Once it has recognized, then the CD4 helper T cells will be activated. Once they've been activated, they will increase in number, they will multiply. We call it clonal expansion. Okay, it's like kufanya cloning. So zinakuwa mingi, many copies of the same. Ni kama zinaitana. Okay, so they make copies of themselves ndio ziweze kusaidia. So these helper T cells tulisema they are not involved in the killing directly, but they release chemical messengers ile tulikuwa tunaita cytokines. Okay? The cytotoxic T cells, the CD8, on the other hand, they will also become activated if the antigen imeshikana na the correct MHC molecule. So they will also become activated. They will multiply. Then they will now be ready to kill that infected cell. Okay? They'll be ready to kill that infected cell or to kill that abnormal cell. So it is important that these T cells are activated because activation ensures that the response is specific. It is responding to a particular pathogen. That is important for memory. Okay? Ukisikia kitu fulani inasema hiyo ni HIV, hiyo ni TB, hiyo ni cancer. Okay? So it has to be activated. Tuliona activation, recognition is only possible when the T cell has been activated. Okay? How does it recognize? How is the activation? When the antigen imeshikana na the correct MHC. Specific. Particular antigen, correct MHC. Very specific. So activation will ensure that the response is very specific. Then activation will also ensure that there is a strong immune attack. That's why multiplication is happening. Okay? So once the T cell has been activated, then it multiplies, increases in number, so that when it attacks, that attack will be strong. Then activation also ensures that the action is targeted to that particular pathogen, targeted to that particular cell. Okay? There was recognition, there was presentation, there has been activation. Now, the immune attack happens. So after activation, then the cytotoxic T cells, for example, they can now kill. The cytotoxic T cells, they are the CD8. How do they go about the killing? First, they have to attach to that infected cell, or they have to attach to that abnormal cell. After they have attached, we saw that the cytotoxic T cells will kill a cell using two main powerful molecules. One, they release the perforins. Kazi ya perforin is to perforate, kutengeneza mashimo on the cell membrane. Then kupitia kwa hiyo mashimo, the granzymes which will be released by the cytotoxic T cell itapita kwa hiyo mashimo. The granzyme ikishapita itaingia kwa hiyo cell, ikifika ndani ita trigger the killing of that cell. Okay. Cytotoxic T cell, it attaches to a cell which is infected. It attaches to a cell which is abnormal. After attaching to that cell, it releases two powerful chemicals. Perforins, which will perforate. Perforating ni kuweka mashimo. It now releases granzymes. Granzymes zitapita kwa hiyo mashimo yenye perforin imetengeneza, ingie ndani ya the infected cell or the abnormal cell. Once the granzyme is in, it will activate enzymes which will now trigger the death of that particular cell. Okay? The death of the cell is what we are calling now apoptosis. So the result of that ni kwamba the cell that is infected will die. The cell that is abnormal will die. Once that happens, then the infection in that cell will not spread. Whatever is making that cell to be abnormal will also not do what? Spread. Okay? So, helper T cells can also help to strengthen the response. Okay? So, how do they aid in ensuring that the response is stronger? They lead B cells to produce antibodies. The helper T cells also increase the activity of the macrophages. Okay? The helper T cells also recruit neutrophils and other immune cells. The helper T cells will as well enhance the activity, the cytotoxic T cells will make their activity more efficient. The cytotoxic T cells kazi yake ni kuua. So it means the helper T cells zitasaidia the cytotoxic T cells to kill the infected cells, the abnormal cells better. They kill them more efficiently. Okay? So without these helper T cells, the immune response will become weak and will become uncoordinated. So it's important that these helper T cells are also present in an immune attack, so that they help in ensuring that the immune response is strong and the immune response is properly coordinated. Then the fourth step is now in the formation of the immune memory. After the infection, after the infected cell, after the abnormal cell has been cleared, then some of the T cells will not die. Okay? Some of the T cells that helped in the process, they will not be killed. Not all of them will die. Some will hang around within the body of that particular organism for a given period of time. By hanging around, they've now become what we call the memory T cells. So the functions of these memory T cells, that in case there is a reinfection is an infection by the same pathogen. So you had For example, Mycobacterium TB attacking you today. The T cells fought it and the infection is cleared. Some of those T cells that helped in that process of clearing the Mycobacterium TB, some of them will not be destroyed. Instead, they will hang around and continue being part of the memory T cells within your body. So in case in the future you encounter the same Mycobacterium TB infection, these memory T cells, which were kept from the previous infection of Mycobacterium TB, they will respond immediately because they know, okay? So because of that, then the immune response for the second infection will be faster, will be stronger, and will be more effective, okay? So this immune response that has been brought about by the memory T cells is what we call secondary immune response. Is a secondary immune response because you are using cells that were created earlier. When you had the first infection, these T cells were not cleared from the system. Zilibaki. So when you now have the second infection, the secondary infection, these T cells have brought up a coordinated immune response. That's why we are calling it the secondary immune response. So just to put things together, when a pathogen enters the body, a pathogen is an infectious agent. Once it enters the body, it enters the cell, for example. That pathogen will be broken down. Then the antigens will be presented using the MHC. Okay? So once the antigen has combined with the correct MHC, then T cells will be able to recognize that particular antigen. Once the T cells can now recognize the antigen, then they will be activated. Upon activation, various types of T cells zitaanza kufanya kazi yake. When the T cells are activated, the cytotoxic T cells zitaua the infected cell or the abnormal cell. The helper T cells zitasaidia in the coordination of the immune response. The regulatory T cells, they will ensure that there is no overreaction. The immune system is not working in excess of what is expected. Then the memory T cells zitahakikisha kwamba some of them are kept so that in case there is a future infection, these memory T cells will offer future protection, what we are calling secondary immune response. Okay? So, in summary, we have helper T cells, ambaye kazi yake is to activate other immune cells, helping in the coordination of the immune response. The cytotoxic T cells, kazi yake is to kill either the infected cells or the abnormal cell. Regulatory T cells, kazi yake is to regulate the immune system, to ensure that the immune system does not overreact, so it suppresses the immune response. Memory T cell, kazi yake ni to provide long-term protection. They hang around so that in case there is a reinfection, then these memory T cells are present to offer that protection. We call that secondary immune response. Awesome. Okay. On page 13, I provide you with the diagrams just to help you know the flow. Okay, how cell-mediated immunity works, how, for example, the immune coordination works, and how the memory situation forms. Okay, so you just have these flow diagrams to help you. You have these flow diagrams to help you when you are thinking through after you've read and watched this video. Okay? So, up to that particular point, we've dealt with the T cells, what they do. We've looked at the MHC to some extent. Okay? I'll stop there. Then in my next video, I'll begin dealing with what exact role the T cells play in offering protection against infections. Okay. Then from there, from that particular point on, we'll continue looking at other aspects. Otherwise, have a look at this, read through the notes, then if you have a concern, if you have a question, do not hesitate to reach out to me. Look out for the next video when you'll be looking at the exact role that these T cells now play in protecting against infections. Thank you so much.